Imagine a microscopic battlefield where tiny invaders fight for survival. On one side, you have bacteria. Euros contain cells with walls, ribosomes, and the ability to divide on their own. On other, viruses. Simple protein shells that hijack host machinery to replicate. Today we dissect how these two types of pathogens differ at every mechanistic level. Viruses are submicroscopic particles that lack the cellular machinery required for independent metabolism. Bacteria, in contrast, are single-cell organisms with a complete set of organelles and a defined cytoplasmic membrane. The most striking size difference is that viruses range from 20 to 300 nanometers, while bacteria typically span 0.5 to 5 micrometers. A virus, Eurostraid's outer shell, called the capsid, encloses its nucleic acid but offers no metabolic enzymes. Bacterial cells possess a peptidoglycan cell wall that provides structural integrity and defines their shape. Some viruses acquire an additional lipid envelope derived from host membranes. Bacteria do not have such envelopes. The genetic material of viruses can be DNA or RNA, whereas bacterial genomes are always double-stranded DNA. Replication of a virus requires hijacking the host cell Eurostraid's ribosomes and polymerases to synthesize new virions. Bacteria replicate autonomously through binary fission, duplicating their chromosome and dividing without external help. These structural distinctions underpin the different mechanisms by which viruses and bacteria interact with living organisms. 0.8. Bacterial cells are bounded by a rigid cell wall that provides shape and protection against osmotic stress. The wall is composed mainly of peptidoglycan, a polymer of sugars and amino acids arranged in a mesh like lattice. Inside the membrane lies cytoplasm, a jelly like matrix where metabolic reactions occur. Ribosomes scattered throughout the cytoplasm translate messenger RNA into proteins, essential for cell function. In contrast, viruses lack this complex internal organization. They are essentially nucleic acid surrounded by a protein shell. The viral genome can be either DNA or RNA, singly Euro-stranded or doubly Euro-stranded, and carries all information needed to hijack host machinery. Encasing the genome is a capsid formed from repeating protein subunits that provide structural stability. Some viruses also possess an outer lipid envelope derived from the host cell membrane, but it is not part of the core architecture. Because viruses do not have ribosomes or cytoplasm, they cannot carry out metabolism independently and must rely on infected cells for replication. The stark difference in cellular components explains why bacteria can survive freely while viruses require a living host to propagate. 0.8. Bacterial cells begin replication by duplicating their circular chromosome and partitioning it into two future daughter cells. The process is initiated when a single origin of replication is recognized by initiator proteins that unwind the DNA helix. Once unwound, DNA polymerase Roman III synthesizes new strands in both directions, creating two identical copies. After synthesis, the cell elongates and forms a septum that divides the cytoplasm into two separate compartments. In contrast, viruses lack their own replication enzymes and must co-opt host machinery to produce progeny genomes. Attachment begins when viral surface proteins bind specific receptors on the host membrane, triggering conformational changes. The virus then injects its nucleic acid RNA or DNA directly into the cytoplasm, bypassing the nuclear envelope in many cases. Host RNA polymerase Roman II or reverse transcriptase is recruited to transcribe viral genes, while viral encoded polymerases replicate the genome. New viral genomes are assembled with capsid proteins and released when host membranes fuse or burst, completing the cycle. Comparing these mechanisms reveals that bacterial binary fission relies on intrinsic enzymes, whereas viruses depend entirely on hijacked host processes. 0.8. Bacterial attachment begins when pili extend from the cell surface and latch onto specific glycans on epithelial cells. Flagella, while primarily motile structures, can also contribute to adhesion by creating localized shear forces that bring bacteria closer. Once attached, many gram-negative species deploy a type Roman III secretion system that injects effector proteins directly into the host cytoplasm. These effectors can manipulate signaling pathways to suppress immune responses or promote bacterial uptake. In contrast, viruses rely on surface spike proteins that recognize and bind specific host receptors such as ACE2 or CD4. Binding triggers conformational changes that expose fusion peptides, allowing the viral envelope to merge with the cell membrane. Alternatively, some viruses are internalized through receptor-mediated endocytosis, where the host vesicle engulfs the virion. Once inside, the viral genome is released into the cytoplasm or nucleus, initiating replication. The juxtaposition of bacterial pili and viral spike proteins illustrates how different pathogens have evolved distinct entry strategies. Understanding these mechanisms provides a foundation for designing interventions that block attachment or fusion without invoking clinical claims. Pause 0.8. Pattern recognition receptors are the first line of defense that detect conserved microbial signatures. Tola-urose-like receptor 4 TLR4 sits on the cell surface and binds lipopolysaccharide LPS from Gram-urose-negative bacteria. When LPS engages TLR4, adapter proteins such as MyD88 recruit Iraq kinases to initiate a transcriptional program. In contrast, bacterial peptidoglycan is sensed by intracellular receptors like NOD1 and NOD2 that activate N5-urose I-circumflex OB signaling. Viral nucleic acids are recognized by cytosolic sensors. Riga urose I detect short 5A circumflex urose UA circumflex urose triphosphate singlia urose stranded RNA ssRNA. MDA5 prefers long W urose stranded RNA dsRNA, and both receptors trigger the production of Ti by interferons. The downstream cascade involves phosphorylation of IRF3/IRF7, leading to transcription of antiviral genes. These pathways illustrate how innate immunity discriminates between bacterial and viral threats through distinct molecular patterns. The signaling networks converge on shared transcription factors, but produce different cytokine profiles tailored to the pathogen type. Understanding these mechanisms clarifies why certain infections elicit strong inflammatory responses, while others trigger antiviral states. Pause 0.8. Bacterial pathogens often rely on direct physical touch to move from one host to another. When a person touches a contaminated surface, bacteria can adhere to the skin and later be transferred to mucous membranes. The Handa-urose-Toa-urose phase transfer is a common route that bypasses environmental barriers and delivers microbes directly into entry portals. In contrast, many viruses are released into the air when an infected individual coughs or sneezes, forming droplets that travel short distances. Smaller aerosolized particles can remain suspended for extended periods, spreading through ventilation systems or open windows. Droplet transmission is limited by gravity and typically requires close proximity to the source. Some viruses exploit arthropod vectors. A mosquito bite injects viral material directly into the bloodstream of a new host. The vector pathway bypasses external surfaces entirely, allowing rapid dissemination across geographic regions. Visualizing these mechanisms helps clarify why hygiene practices differ for bacterial versus viral threats. Understanding the distinct pathways informs targeted prevention strategies without implying medical interventions. Pause 0.8. Bacterial spores are dormant forms that encapsulate the cell in a multilayered coat composed of proteins and peptidoglycan. This coat contains small acid-soluble spore protein SASTs that bind DNA tightly, shielding it from ultraviolet light and chemical damage. Because of these structural features, spores can survive extreme temperatures up to 120 A circumflex degrees C for hours and pH ranges from 2 to 12 without loss of viability. In contrast, viral capsids are protein shells that protect a single-stranded or double-stranded nucleic acid core. Capsid stability depends on the number of disulfide bonds and the presence of stabilizing ions. Most capsids remain intact at temperatures up to 60 A circumflex degrees C. However, many viruses lose infectivity when exposed to pH values below 4 or above 9, because their capsid proteins unfold and the genome is degraded. The resilience of spores is further enhanced by dehydration, which reduces water activity and limits hydrolytic reactions that could damage macromolecules. Viral capsids lack such a dehydrated core. When the surrounding medium dries, capsid proteins can aggregate or lose their native conformation. Therefore, environmental stability assays often show bacterial spores surviving for months in dry soil, whereas many viruses persist only days under similar conditions. These differences highlight the importance of spore coat chemistry versus capsid protein architecture in determining how long a microorganism can remain viable outside its host. Pause 0.8. We compared the fundamental structures of bacteria and viruses, examined their distinct replication mechanisms, explored how they interact with host cells, highlighted the innate immune sensors that detect them, and discussed differences in transmission routes and environmental stability.